Okay, that last bit about speciation in flounder taking less than 8,500 years raises a question as to how fast it happens. And this actually cuts back a bit to Darwin and his thought that evolution always took place by very, almost indetectably tiny steps over extremely long periods of time. We can look at speciation through looking at what are called adaptive radiations. And these are cases where you have a founder species that encounters a new habitat, often but not always dispersing to a new habitat, encounters a lot of unoccupied ecological niches, and radiates pretty rapidly into a large number of closely related species. The result is what we call a species flock. And the classic example would probably be the Galapagos finches, because here you have 14 species of finch, although technically 13 are in the Galapagos, and one is a little bit to the north on another tiny little piece of rock called Cocos Island, which presumably the ancestor of which would have dispersed roughly 4 million years ago, and then diversified into what we see now. You can find some other awesome examples elsewhere in the world, and basically if Darwin had known anything about these, he would have written basically the same book, because it's basically the same story. So for example, if the beagle had crossed the equator and gone up all the way to Hawaii, instead of visiting the Galapagos, Darwin might not have written about the finches, but he might have written about the honey creepers. Which is an even bigger species flock with 58 species, although if memory serves, roughly half of those have gone extinct. There's about 120 species of land snails in French Polynesia. Unfortunately, many of those have gone extinct because of an introduced snail. Somebody thought it would be a bright idea to introduce an African species of land snail, which promptly ate all of the native ones. Hawaii is also ground zero for fruit fly diversification in the genus Drosophila and a related genus called Scaptomyza. There's over 700 species. Species can be restricted to different islands, different sides of an island, sometimes even separate valleys on the same island will have different species of fruit flies. And it is presumably just the good old story of, dispersal and separated population speciating. If you're the size of a fruit fly, 2 valleys on a mountainous island may be almost impossible to get from one to the other. You were asked.
What is a honey creeper exactly?
How about I just show you?
Oh.
Okay. There are a group of birds in the family Treponandidae. And Hawaii, bear in mind, is 2,000 miles from the nearest mainland. Even by Pacific Island standards, it's pretty darn isolated. Humans, of course, have brought a lot of species, beginning with the Polynesians, and of course now they're having real problems with introduced species. But the honey creepers are native, and they've radiated into 57 species. I think the previous slide said 58. There may be some debate on the matter. But unfortunately, nearly 2/3 are extinct or probably extinct, 39 out of 57. And it's the same story as the Galapagos. They've got different beak types. The beaks are adapted to different diets. Some of them are adapted to sipping nectar, or some for eating fruit, some for eating insects, some for cracking seeds. And again, it's very much like the Galapagos. You would have had a founder reach the Hawaiian Islands. I don't know what the nearest relative of theirs would have been, but maybe they got blown there in a storm or something. We may never know. Founded a population, and as that population spread, it got subdivided into different populations on different islands, each one of which would have speciated and adapted to local conditions. There you've got, those happen to be 4 of the Hawaiian honey creepers, at least one of which, Chlorodops kona, I think is extinct. I think the iiwi up at the top left is one of the few that is not particularly endangered. And one of the most beautiful birds in Hawaii with that brilliant scarlet color and a song that allegedly sounds like something you'd hear, like a soundtrack, a sound effect you'd hear in an old Warner Brothers cartoon. Now, apparently they sound quite ridiculous, but they've got that very long curved beak. That's for nectar sipping. The Kona grosbeak over there with a much heavier beak, that's a seed cracking bird. I'm not sure what Sagittariostris up there does, the amakihi, but you can see that's a very narrow beak. That may also be a nectar sipper, but maybe different flowers. I think Sitarostra is a fruit feeder, and it's got a big, Sitarostra means parrot beak. So I assume that's got a more parrot-like diet. So yeah, you can see you have this great diversity in a group of birds restricted to a very limited area, and it is probably very much the same story as you get in the Galapagos Islands. Probably the biggest is the cichlids of the African Rift Valley Lake, and cichlids are members of a family of fish, the cichlidae. That helped. You can actually see some of these in any pet store that's got a decent freshwater aquarium department. There are some African cichlid species that are quite beautiful and can be kept by amateur aquarium hobbyists. It's been a minute since I went to PetSmart, but I think the last time I checked, they actually had a tank of selected East African cichlids. These are native to some very large lakes in the East African Rift Valley. I'll talk more about this probably towards the very end, but in the East African Rift Valley, the continent of Africa is very, very slowly splitting apart. Given another few million years, and East Africa is going to be a very big island, basically much like Madagascar is now, except even bigger than that. And in the valley where the continent is slowly pulling apart, we've got several big freshwater lakes. And one of those lakes is Lake Victoria. And until recently, it contained 300 different species of cichlid. Unfortunately, humans introduced Nile perch into the lake. Those are fished for food, and the Nile perch promptly ate most of the unique cichlids like they were popcorn, and probably about 200 species of cichlid are extinct by now. There's a good book came out about 20 odd years ago on these called Darwin's Dream Pond, which you might look at if you have any further interest. There's 2 other major lakes in the East African Rift, especially Lake Tanganyika and Lake Malawi. And in total, there's about 1,000 species in all of these East African Rift Valley lakes. And as I mentioned, you can find some if you follow the freshwater aquarium trade. I don't know about Pet Country, or I don't know about Petco, but PetSmart, at least until fairly recently, used to have some of these. Maybe I'll go by and check sometime. And the species are all different in color, they're different in size, in shape, different in breeding behavior. Let me think, there are some that let their eggs float, some where the male makes a nest and guards them. There are some that are mouth breeders, which means one of the parents just holds the eggs in its mouth and even guards the young in its mouth until they are mature enough to be out on their own. That's South American. And certainly different food supplies. Some of them feed on plants, some on snails. There's at least one species that is lepidophagous, which means it takes bites out of the scales of other fish. You know, it's a living. And those are the lakes. Lake Victoria is up there where Tanzania, Kenya, and Uganda meet. And then Tanganyika is between Tanzania and the Congo. And Lake Malawi is between Tanzania, Malawi, and Mozambique. And here's a few of the Lake Victoria cichlids. Many of those, by the way, are males in breeding coloration. A lot of these will get very bright. The males will get very bright colored, but only when it's time to breed. And the males will actually develop so-called egg spots, those white spots that you see on the anal fin. So the females aren't necessarily this brilliant, and the males may not be as well when they're not in breeding form. But you can certainly see that they have different color patterns, different mouth sizes and shapes, and that's correlated with diet. Cichlids actually have jaw bones that have teeth on them, and these snail eaters would have very big, chunky, crushing teeth plates, whereas insect eaters might have much lighter ones. Different jaw size, different proportion. So that's 6 of the 300 plus species that we have there, or used to. And again, here we've got constraints on how long this took place. And we actually know that Lake Victoria completely dried up 12,400 years ago. That seems like an extremely long time, again, but they were fully modern people hanging around there. Looked not grossly different from us. I mean, probably had darker skin than the people in this room, but you know, would have looked pretty much like modern Africans. They would have been hunters and gatherers, but they had brains as big as ours, and you know, would have had culture and mythology and religion and violence and hatred and all of these things that make Homo sapiens so wonderful. It wasn't that long ago. This is roughly the same time when you have the last ice age gripping Europe. And we know it because if you drill a hole in the bottom of the lake, you get layers of lake silt. But you go below that and there's a layer of soil. Soil that is now completely buried in silt when the lake filled up again. So the 300 species of cichlids must have descended from a common ancestor in less than 12,000 years. So if you divide that, you know, speciation isn't necessarily nearly quite that regular, but that works out to an average of 1 species every 40 years. There's actually a small lake, Lake Nabugabo, that's been separate from Lake Victoria for 4,000 years. And it has 5 cichlid species that are not found anywhere else in the world. So there you've got 5 new species that must have formed in the last 4,000 years. 4,000 that's younger than the pyramids of Egypt. We've had civilization for longer than 4,000 years. 4,000 years ago is, oh, what was going on? Yeah, ancient Egypt is hitting its stride. So is ancient Sumeria. Early Chinese civilization is going on. They're building what looks like an early civilization down in Poverty Point, Louisiana. That's well within the time frame of human experience. And yet there's 5 species in Lake Navugabo that must have evolved there because they're not found anywhere else. What may have happened was if the lake levels ever got low, you know, imagine a lake drying up. Lakes typically don't have very even bottoms. So instead of the whole lake drying up at once, you're likely to get little isolated ponds and puddles, especially around the edges of the lake. And the idea is if the lake gets low, you'll get isolated, you know, little ponds around the rim of the lake. In those isolated ponds, you get new species forming. And then the new species come together when the lake level rises again and joins all the ponds up. That's a case, that's an example of what we call peripatric speciation. Peri just means around, as in perimeter. Peripatric speciation means around the border of the homeland. Yes?
I just want to point out that Mohenjo-Daro was like 5,000 years.
Okay. Okay, thanks for.
The Indus River Valley civilization.
Okay, thanks for adding that. So there was, you know, Egypt's going strong. You had Sumeria, which also had Mohenjo-Daro in India, early China. There's a number of civilizations that have popped up by about 4,000 years ago. You know, I don't know, not as far as I know, not in that far south in Africa, but we've got Egyptian texts that we can read that are older than 4,000 years. Anyway, okay. Lake Tanganyika's held water for over 3.5 million years, and its cichlids are even more diverse in shape than those of Lake Victoria, probably because there's been more time for them to adapt and diversify. So there you've got 6 different Tanganyika cichlids, and it may be hard to tell, but notice that the scale bars for some are 1 centimeter and some are 5 centimeters. So there's a big difference in body size as well as body shape, fin shape, etc. We used to assume with Darwin that except in cases of speciation by hybridization or polyploidy, which only take a generation or 2, that speciation was always this really gradual process. And that tends to be the legacy of Darwin, who got the idea from BIOL that all you need is very slow processes multiplied by very long periods of time. A gent by the name of Richard Goldschmidt in the early 20th century, poor guy, German biologist, before World War II, he's highly esteemed. He's got his own lab. He's got, you know, a high salary, lots of students, but he fled to the United States during World War II and took a job at the University of California at Berkeley, and they didn't have anything else for him to do, so they put him to work teaching freshman biology. So imagine taking biology 1440 with someone who, A, you know, is a truly world-class scientist, but he's totally traumatized because he's been forced to immigrate and brought low, and B, can't speak English. Yeah, it did not go that well. I don't know what your 1440 experience was like, but it could have been a whole lot worse. Could have had Richard Goldschmidt. You know, brilliant man, but probably not best for 1440. Yeah. Except they called it biology 1A at Berkeley. Anyway, sorry about the accent. He proposed, And he'd taken this as an earlier idea. You might remember that people like De Vries and even Huxley played around with the idea that new species could appear rapidly because of saltation, because of rapid, larger scale genetic changes. Goldschmidt suggested that new species might arise by macromutations, which are mutations that would have large-scale phenotypic effects in, that might alter, some, not just some little minor parameter of beak size or something like that, but something like number of legs or something that could give you a radically new body plan. He was probably not right, but as I'll show next week, he was on to something. This is one of those examples of what I call Wagner's law of the history of science, which you all ought to write down because it might well pop up on the exam. Okay, that got your attention. Wagner's law of the history of science states that it is often more fruitful in the long run to be wrong in an interesting way than right in a boring way. And people like Lamarck are a great example. He was wrong, but in such an interesting way that his ideas lasted for a long time. They're actually getting a new hearing, and in some cases, they may actually turn out to be more right than he realized. And Goldschmidt's another example. Goldschmidt's idea was kind of derided and became known as, he did not call it this, it became known as the hopeful monster theory. The idea is that if you get one of these macromutations, it produces, a monster, but hopefully that monster might manage to be adapted even better to the environment that it's in. That's not exactly what Goldschmidt meant, but it kind of got tagged with that sobriquet, that nickname. And as we'll see next week, he was asking the right questions, even if he couldn't quite get to what I think are the right answers. So Goldschmidt has suggested that speciation, even without chromosomal hijinks like polyploidy, speciation could happen fast. The older assumption was that if you looked at the history of a species in the fossil record, most of what you would see is directional trends. In the older rocks, you might have members of the species that were, let's say, small, and they very gradually get larger and larger and larger as you dug in progressively younger and younger rocks. So if you could watch a species evolve, most of what you'd see would be gradual trends within that species or within that lineage. That's called anagenesis, by the way, directional change within a lineage over time. The problem is when you look at the actual fossil record, you don't really see that. And it used to be assumed that if you didn't see that, it was because the fossil record was imperfect. Be it said, the fossil record is imperfect. And the reason I say that is, first of all, we don't have anything like all of the fossils. I mean, we keep finding new species every year. I mean, even in the dinosaurs, there's probably, I'm not even sure, maybe 20, 50 new species of dinosaurs described in a given year. And there's even more new things that we keep finding in, let's say, the marine invertebrates. We have not nearly exhausted the supply yet. And of course, in many living groups, there may be almost no fossils. There is a major phylum called the Rotifera that's very ecologically important in the oceans and in fresh waters. The total number of fossil instances that I know of is exactly 3. And I should know because I discovered one of them. So you are hearing the words of the man who discovered 1/3 of the fossil rotifers currently known. This is your cue to go, ooh. Ah. All right. Okay. I've got tenure too. All right. Yay. Flatworms, things like the planaria worms that you've probably seen, as well as a lot of important parasites, very important ecologically, probably go back hundreds of millions of years. Total number of significant fossils that are any real age is, I believe, one body fossil and some indirect evidence of parasitism that is basically funny pits on inside seashells. So this is a huge and very important group, and we know almost nothing about its history from the fossil record. Even in better represented groups with hard parts that do fossilize, like mollusks or heck dinosaurs, we keep finding new ones. And one of my old professors, Jim Valentine, estimated that if you looked at just any typical shallow water marine habitats, if you go to a beach and you counted up all of the species that were present, only about 13% of them would normally form fossils, would have shells or what have you that were beefy enough that they had a good chance of surviving after getting buried. 5/6 of the life at one of your typical beaches is so squishy, it doesn't have any hard parts, and it's very unlikely to form recognizable fossils at all. So it is true that the fossil record's very imperfect. And so this classical model of evolutionary change through time is a population has some trait, it's got a bell curve distribution, and some members of the population have a very small whatever it is, some have a large whatever it is, and most are in the middle. And as you go through time, going from bottom to top, that distribution just slides in one direction over time. And again, that's anagenesis. This, by the way, would also presumably be the result of directional selection. Remember, that's what you get when you have some trait plotted on a bell curve, and the bell curve slides to the left or slides to the right. The mean shifts over time. Remember, right? Directional selection. What's that?
I was going to say not stabilizing.
Not what?
Not stabilizing.
Not stabilizing and not disruptive, right? So the old model was that if you had a really complete record, you'd get something like this. The mean of that trait would slowly shift in one direction and gradually shift. There's no zigs and there's no zags. And that would be anagenesis. There's some data, and well, it's not really data because of the way it's drawn. You're not really seeing a bell curve. And the arrows have been drawn to make it look like this is gradual change, but really the fossil record is not fine-grained enough to be sure of that. But this is the kind of thing that you would expect to find. Those what's labeled fossiliferous units on the left are just rock layers in Texas, from oldest on the bottom to youngest at the top. Starting at the bottom with the Wilcox group and ending up at the top with, just a group of fossil-bearing rock layers called the Jackson group. And up there at the top right, Athleta petrosa symmetrica, that's a fossil from the Jackson group. And here's another fossil from the Jackson group. A, B, C, easy as 1, 2, 3. Simple as do and mi, A, B, C, 1, 2, 3, you and me, girl. Get it? Jackson group, right? Fossil. Okay, sorry. I don't get to make that joke very often. Besides, look, I got the high notes, you know, yeah, I've still got my falsetto. Whatever it's worth. Anyway, the way this is drawn, this looks like it's showing you a trend towards the spines getting gradually longer and longer, and the size getting gradually bigger and bigger. Now, the arrows right there are not data, and we don't really have enough fine-grained data to really see if this is as gradual a transition as it seems, but this is what people expected to find. You do have some more fine-grained data here. What you're looking at here is data from a shelled protist called a fusillinid foraminiferan, species Lepidolina multiseptata. Don't worry about what this thing is. It's a single-celled critter, but it makes a rather complicated calcium carbonate shell around itself. These can be extremely abundant in certain rocks. My major professor was an expert on these protists, on foraminifera, basically because you can find fossils of them anywhere, and they're found in all the world's oceans, which basically means you can write grants to travel anywhere you want to go in the world, because no matter where you go, there will be living or fossil forams. And that's why my major professor had visited something like 150 countries in his lifetime. And, you know, knocked up women in most of them, but that's another story. Yes?
Is it coral reef and calcium carbonate?
Yeah. And these are not coral animals, but you do find foraminifera on coral reefs. But I mean, you can also find them in the Antarctic. My major professor ended up with a small island off the coast of Antarctica named for him because of his participation in a major, you know, National Science Foundation funded research expedition to Antarctica. Anyhow, so this is some data from fossil members of the group. And the y-axis right there is time. And again, those things that say Sisyphon, limestone, C member, D member, that's just different rock layers. Oldest on the bottom, youngest on the top. And what you're looking at is measurements of a portion of these protist shells, a portion called a proloculus. And each one of those bars gives you an idea of the variance and the standard deviation in each sample from a different level in the rock stack. And in the center of each bar, that's the mean. So at the bottom of the rock stack, the shell part averages 200 microns in diameter. By the time you're at the top of the rock stack, it's averaging 550 microns. So it's about over 2.5 times as large. And you can see that there's a fairly steady trend towards larger size over a period that's probably about 15 million years. The shell protist just keeps getting bigger and bigger and bigger. But at a very slow rate, gaining, I don't know what the average might be, but maybe, I don't know, 10 microns every million years, 20 microns every million years, something like that. Enter a guy named Stephen Jay Gould at Harvard, a specialist in the fossil record of land snails. And his buddy Niles Eldridge, a specialist in a group of extinct arthropods called trilobites. They argue that if you take the fossil record seriously, you don't really see sustained anagenesis like that. That type of gradual plodding change with no zigs and no zags is not really the way that evolution happens. what you get instead is a pattern that they refer to as punctuated equilibrium. If you like, you can call it punk-eq if you don't feel like writing out the whole thing, punctuated equilibrium. And in punk-eq, when you look at the fossil record through time, new species appear abruptly. Now, be careful with this because I get this from the creationist side every so often, that when we say abruptly, the claim is that we mean instantly. And the answer is no, it's not instantaneous. It's just fast relative to the total length of time that a species is around. And the species will then stay very similar, much the same, maybe not exactly the same, but it will stay fairly steady for maybe a couple million years. So punctuated equilibrium does not mean that species appear in an instant. It means the length of time that it takes for them to appear is relatively very short compared to the lengths of time that they are in fact around. Now, we've already seen some examples of speciation where the time frame is a few thousand years, right? Like the Lake Nabu-Gabo cichlids or the Baltic flounder. We've seen partial speciation taking place over just a few hundred years. So if we say that, if we give it a very generous estimate and say that it might take 10,000 years for a new species to appear, 10,000 years is still maybe 0.5% of 2 million years, which more or less might be about the amount of time a species might be expected to exist. Right? So 10,000 years in human standards is incredibly long. You know, 10,000 years ago, our distant ancestors were all running around with spears hunting. Well, the woolly mammoths had pretty much died out, but they were hunting something. You know, that's far older than human civilization. Compared to the total length of time a species might exist, that's 0.5%. You know, that's not very much time at all. So the time it takes for a species to appear is very abrupt compared to the length of time a species might persist without very significant change. And the predominant evolutionary dynamic is not anagenesis, it's cladogenesis. And again, klados happens to be ancient Greek for a branch. Cladogenesis is the branching off of new lineages. And that branching off takes relatively short periods of time compared to the total length of time that the species is around. Punk-eq often gets confused with the hopeful monster theory, but they're not the same thing. Punk-eq is not a theory of how species form. Although Gould ended up coming up with what he hoped was a comprehensive theory of how species form, and it's over 1,000 pages, so dang. Punk-eq is a description of the pattern of species formation over time. And when you're talking about whether new species appear gradually or in punctuation, you always have to keep a relative time scale into account. It's relative to what? Long time ago, I was kicking back in my apartment, you know, probably just drinking a beer and watching the game one afternoon, and there's this knock on my door. And I walked over to see who it was, and there's this snail on my doorstep. And the snail says, "Excuse me, sir, if you have a moment, I'd like to speak to you about Amway products." I think to myself, good grief, it's a door-to-door salesman snail. I don't have time for this stuff. So I just reared back and kicked the snail into the bushes and slammed the door. Okay, 3 years later, I'm in my apartment and the door, I hear a knock on the door again, and I open it and the snail says, "Hey buddy, what was that for?" Get it? My point, to the extent I have one, is that when you're talking about whether something is happening quickly or gradually depends on your frame of reference. which is kind of like the one about the snail who comes crawling into the police station and says, "You got to help me, I just got mugged by a couple of turtles." And the cop says, "Can you describe them?" And the snail says, I don't know, it all happened so fast, right? Whether something is fast or slow depends on your perspective. What is fast to a snail is not fast to you. So in a gradualistic model, you do get new species branching off, but change is relatively slow. In a punk-eek model, change is concentrated in these relatively brief bursts of time, and for most of the time that a species is around, it doesn't change form very much. Hence the name punctuated equilibrium. The equilibrium part is the million or so years during which a species doesn't really get altered radically. And the punctuated part is the brief period of time where most of the change is actually happening. Let's look at some actual data. This was some work done by a guy who, last I heard, was at City College of New York, living in New York City. His name was Ross Nain. It's funny, he actually thought about applying for this job that I currently occupy when we were both in grad school, and decided not to apply because, quote, "I don't think I can handle the South." So I got this job, and poor him has to live up in New York City with, you know, all the symphony and the cultural attractions and amazing ethnic food. Poor guy. I feel sorry for him. I do.
Yeah, good for him. We don't work here anyway.
Yeah, well, he could be something of a Debbie Downer. Behind his back, I used to refer to him as Eeyore. Yeah, well, something like that. Yeah. Although he did have reasons for being a Debbie Downer, because one year he got hit by a car and ended up in the hospital, and the car was being driven by an off-duty Marine. So he got visits from the guy's commanding officer trying to threaten him into not filing a lawsuit. So, you know, Marines. Anyway, so when Debbie Downer was not moaning about how he couldn't handle the South, and when he was not getting run over by cars, he studied a group of marine snails called marginellids, and this is a couple of marginellids. They're not really big. They're attractive enough. You might sometimes find them used in shell jewelry. So you'll find marginellids if you ever buy cheap shell necklaces in gift shops in Hawaii or Florida or something like that. But they do have a pretty extensive fossil record. And he went on down to Costa Rica and made a whole bunch of measurements on a lot of fossil marginellid shells from Costa Rica. This is his diagram of the measurements that he made. And he combined these measurements into a single number using principal components analysis. And I mentioned this earlier. This is a way that you have where you can take many different measurements on a set of things and boil all those measurements down into a single number that expresses the largest possible fraction of the variation in the sample. So instead of trying to graph height, width, length, aperture height, spire height, all at the same time on 5-dimensional graph paper, which is kind of hard to get, 5-dimensional graph paper, you can mathematically combine them and get out a number called the first principal component, which captures the largest possible amount of the variation in all of those at once. So he did this, and that's what he actually got. On the y-axis, that's a diagram of the rock layers in Costa Rica. On the x-axis, he's labeled that discriminant axis. That's the same thing as the first principal component. And suffice it to say that if you're a snail with a high value of the first principal component, you tend to be broad. And if you have a low value of the first principal component, you tend to be slender. And in the earliest rock layers, you have, I mean, it's not all that smooth, but you do have a more or less bell curve distribution. In this broad species called Prunum coniforme, it doesn't change very much until you get past sample D. And in sample E, the principal component values slide to the left. That means that the shells are suddenly getting a lot more slender. And by the time you get up to sample H, most of the shells are slender and referred to as a new species called Prunum christine-ladae. There are some broad ones that survive, so it looks maybe like what happened was that coniforme budded off a new lineage that became the slender christine-ladae, but some of the ancestral ones survived, so we might have a split here. And here's the thing, is this gradual or is it punctuated? Well, notice that most of the change is happening in layers E and F. And that's a minority of the total thickness of rocks that's being represented here, right? So you could argue this is punctuated. It still looks gradual, but in this case, we've got a detailed enough fossil record that we can see some intermediate stages in what normally might be too brief an event to be sure that we can capture in the fossil record. There's a limit to the, well, we call it time resolution. Just as you can't time a stopwatch, come on, Wagner, just as you can't time a track meet with a calendar, you can't resolve narrow intervals of time in most of the fossil record. So normally that speciation might be happening fast enough that we can't be sure we can capture it in the fossil record. Here we have, and the transition is gradual, but the transition takes a fairly small length of time compared to the total thickness of the rocks that we've got. Okay, so you could argue this as supporting evidence for punk eke. We can see what the transformation looks like, and it's not instant, but it doesn't take very long compared to the total length of time that these species are around. We can do a little bit better with the fossil record of foraminifera. I mentioned these guys earlier. That's a picture of what one of them looks like. They are marine protists. Some are planktonic, meaning they float, and some are benthic, meaning they live on the bottom. The bodies are somewhat amoeba-like, but they secrete this shell-like covering around the main body of the cell. It's actually called a test. Nothing to do with exams, by the way. The Latin word testa means a pot. So a test is sometimes used for a biological structure that looks like a pot. By the way, the Latin for a little pot is testiculus. Yeah, testicles means little pots. Now you know. Anyway, typically the test is made of calcium carbonate. Sometimes it's made by gluing sand grains together. There's a few other options. It all depends on what foram you're talking about. And the test is shot full of tiny holes that you can barely see in that picture. And thread-like extensions of the cell come sticking out of those holes. The holes are called foramina. And foraminifera means the things that bear holes. That fera means to carry or to bear. Foraminifera are the hole carriers. By the way, I don't know if anybody wants to go to med school, but if you study anatomy, you will encounter foramina. Your foramen magnum is where your spinal cord comes out of your skull. And you've got a bunch more foramina in the base of your skull for the cranial nerves. So at some point you might have to learn the foramen rotundum and the foramen ovale and the foramen lacerum and a bunch more that I actually used to know. The reason why you want to study forams is they're extremely abundant in the oceans. You can find them just about everywhere. My major professor visited 150 countries in his career, going after forams. And in the ocean, the planktonic or pelagic forams, the ones that float, they constantly live, feed, reproduce, die. That's basically life. You ever heard the 4 F's of life? The 4 things that make up the fundamentals of biology: fighting, fleeing, feeding, and sexual reproductive behavior. And these forams fight, flee, feed, and reproduce, and eventually die. And those tests drop to the bottom. There are parts of the oceans that are blanketed in many feet of a mud that's made up of nothing but foram tests. These are roughly the size of a grain of sand. And you get these deposits called foram ooze. And the upshot is that as these forams drop down and die, older forams are constantly getting blanketed by newer ones. And if you take a core sample of these sediments, you can trace evolutionary change in very, very fine detail indeed. And there's some data, and in this case, we've actually got pretty solid estimates of age in millions of years. That's what you're seeing on the y-axis there. And they're looking at the size of the test of a foram species that happens to be called Globirotalia. And it's pretty small, 10.5 million years ago. It stays small until about 5.5 million years ago. And at 5.5 million years ago, the size suddenly goes zigzag. Right where it's labeled transition series, you go from pretty small forams to rather abruptly much bigger ones. And so here we finally got data that's got good age constraints on it. And we see a pretty rapid transition over, in this case, about 500,000 years from Globirotalia plesiotumida to Globirotalia tumida. We've actually captured it this time. And the change only took about 500,000 years, which sounds like a lot, but it's not much compared to, the 5 million years that Globirotalia tumida has actually been around. So again, we have that change, and if we have a detailed enough fossil record, it looks like it's gradual. It would certainly look gradual if you lived through it and you were watching it happen in real time. But it only took about 500,000 years to happen, which is about 1/10 of the length of time that each one of these species has existed without too much alteration. That's what punctuated equilibrium looks like. Speciation takes relatively short periods of time, and then species tend to persist without major change for longer periods of time. And this is actually perfectly consistent with population genetics. In a small population, you get rapid change because it takes little time for mutations to appear, and genetic drift is much bigger. In a large population, you get little drift. It takes a very long time for new mutations to propagate. You get less change in a huge population than you do in a small one. And I think you should have already seen that in one of the labs that you've done. Okay, let me see. So yeah, the transition taking 10 to 100,000 years, if you look at the fossil record, that's a brief instant, and it's often so fast that you can't track it to the fossil record. We're lucky to have a very detailed fossil record of forams. That's kind of like having high-speed film that you can analyze frame by frame. Whereas most of the fossil record is more like having a partial run of a newspaper, where at best you can analyze change day by day, sometimes not even that much. But from the point of view of a worker on modern organisms, that's unbelievably long. 10,000 years is older than human civilization is. And so part of the resolution is that whether a pattern is truly gradual or whether it's punctuated. Sorry, I was just checking time. Depends on the observer's choice of the time scale. Last little bit of a rant. Anagenesis is what gets stereotyped as the way evolution should look. This is a famous diagram from Time Life books in 1968 called the March of Progress. You know, starting with, you know, there's Oog, the ape man on the far left, you know, right next to Gorgo, the Dryopithecus, and Borg, the Ramapithecus. This is a fold-over diagram, so there's a few intermediates that are missing. But then we've got Homo erectus and the guy with the spear. Well, actually, that's a Neanderthal, and then the guy with the spear is a Cro-Magnon. And leading the March of Progress is Fred, the naked white guy, because naked white men are, of course, the pinnacle of evolution. And they're also the people that tend to make drawings like this. And this has been extensively parodied ever since. There's you can see it on the cover of an album by the rock group Supertramp called Brother, Where You Bound? That was a popular meme about 20 years ago, where you had the transition from, you know, chimp to ape man to Cro-Magnon man, which then promptly degrades as you end up, you know, hunched over in front of the computer. You can tell that's an old meme because the computer's got a cathode ray tube screen. You know, these days you'd be hunched over even more tightly, gazing at your phone. So you've seen this a lot, but in human evolution, as in the evolution of everything, the predominant dynamic is cladogenesis. There really is no way you could take all of the fossil record of human relatives or the fossil record of anything, you know, horses, dinosaurs, sporiferid brachiopods, comatulid crinoids, anything. You can't take that fossil record and put all the fossils in one neat chain of progression from beginning to end. Evolution simply doesn't work that way. This came out maybe 10 years ago, and there's actually things we have to add to it because we've discovered new species since then, but it will still give you the flavor. Each one of those darker blue bars represents a length of time that a fossil human relative was around that we know of. And what I want you to see is that instead of a single progression, you get a branching diagram. One of those branches may end up having to be moved backwards in time a bit. I was hearing that just this morning. There's some new species that have been found, and we have to try to figure out where those go on the tree. But this is fundamentally the way evolution works. Evolution branches. Evolution makes trees. And that's actually a pretty good place for a break. I've got 11:13. Let's reconvene at 11:25. And I want to start talking, maybe I can start talking about plate tectonics, which would be really cool. And at the very end, I can hand back the exams of our 3 lucky winners. Everybody who's not here is just going to have to wait that much longer. So I'm going to stop the recording.